Alright guys, so continuing with chapter 9, this is part 3. Alright, so our goal for today is we want to begin to look at the anatomical structure inside of the skeletal muscle fiber. And so we want to start with this organelle called the myofibril. So the myofibril is a special organelle. It wasn't in chapter 3 when we learned about the nucleus and the Golgi apparatus. This is an organelle that actually contracts, and when it contracts, it causes ultimately the skeletal muscle organ to contract or get shorter. That pulls on the tendon is attached to bone, and this is how human beings move their body, okay? So the myofibril is going to be this contractile organelle, and they're only found in muscle tissue. So, get the laser pointer. Where we left off in part 2 was we saw this image, and when we were looking at the myofibrils, they appeared as these little red dots within the skeletal muscle fiber. But it's important to realize that a myofibril isn't just a circular organelle. It's actually cylindrical. So if we come up here, you're looking at part of a skeletal muscle fiber. So you can see the endomysium, sarcolemma. Then you can see all those myofibrils, which again are the little red dots in this cartoon. But here they've pulled out one myofibril, and this allows you to appreciate that a myofibril is actually long and cylindrical and runs the length of a skeletal muscle fiber, okay? And there's a lot of them, so there's not just one myofibril. There's potentially hundreds of myofibrils in a skeletal muscle fiber, and it's that anatomical structure and physiology of the myofibril that we are going to be looking at a lot in the coming slides. Okay, so before we get started with this slide, this second image down here is a potential anatomy short answer for exam 3, so you may want to put that into your notes or put a star beside it right now, okay? So when we're looking at this particular image, there's two terms here that we need to address. So let's start back up here with our skeletal muscle fiber. We've got our nuclei, we've got the sarcoplasm, which is the cytoplasm, and you've got the sarcolemma, which is the plasma membrane, and then you've got your myofibrils, okay? So in the lower part of this image, you can see that the myofibrils have been cut off along the plasma membrane, the sarcolemma. But then up here in this white rectangle, they have left some of the myofibrils so you can see a little bit more detail. And it's the drawing in the white rectangle that we are now zooming in on so that we can see even more detail. So let's start with the things that we've already learned. We know the sarcolemma is the plasma membrane. Mitochondria produces the ATP. Over here, sarcolemma just happens to be labeled twice. Sarcoplasm, again, is the cytoplasm. These myofibrils are cut off, again, flush with the sarcolemma. And then we have several myofibrils that they've drawn out a little bit more completely so that you can see more details. And then here you've got one myofibril that's pulled out even further, okay? All right, so then let's start with the idea of a T-tubule. So a T-tubule is invaginations of the sarcolemma that run between each pair of terminal cisterns. So terminal cisterns is a term that we'll get to in a few minutes. Let's focus on the idea of a T-tubule. So what does it mean to be an invagination of the sarcolemma? Well, so we know where the sarcolemma is. And if you look at this little cartoon, you can see there are these holes, which are the invaginations. There's a hole here, there's a second, a third one, and then coming down the side, there's a fourth, fifth, sixth, so on and so forth, okay? So you've got these sarcolemmas, but then in various places, there are these invaginations, which means that the sarcolemma basically like digs inwards towards the more internal parts of the cell. But that creates basically these tubes, this network of tunnels, and in yellow you can see that you've got this network of tunnels. It's pretty extensive. It actually starts to wrap around the myofibrils, goes to the most internal parts of the skeletal muscle fiber. Well, that network of tunnels that is colored yellow in this picture, those are the T-tubules. And again, the T-tubules are made up of sarcolemma. So use your imagination. Pretend the laser pointer in red, pretend that was a little person, and that little person is just walking around on the sarcolemma and then comes across this opening, this invagination, and starts walking down inside that little tunnel. So you've got this little person who's walking around inside of this little tunnel. Now, again, if you use your imagination and you've got this little person walking around inside of this T-tubule, that person's feet technically are still on the sarcolemma because the T-tubule is made up of sarcolemma. If this little person reached their hands out and touched the side of the tunnel, the walls of that T-tubule, technically that person's hand would be touching sarcolemma. Or if that little person in the T-tubule looked up at the top of the tunnel, that person technically would be looking at the sarcolemma. And so that little person could walk around and get into the deepest parts of the skeletal muscle fiber, but then that person could also walk right back out onto the surface, okay? Why does such a network exist? Well, if you remember, skeletal muscle fibers are voluntarily controlled. That means that technically your nervous system, you can generate a signal from the nervous system voluntarily that reaches the skeletal muscle fibers and instructs them to contract. If you look at this from a big picture point of view, basically what happens is a signal will come in from the nervous system and it ends up being released on the sarcolemma. That signal from the nervous system then ultimately travels down all of these T-tubules and eventually will reach the myofibrils, because you can see that the T-tubules are wrapped around the myofibrils in various places. When the myofibrils receive that signal, they start contracting, and so then when the myofibrils contract, the skeletal muscle fiber will contract and get shorter, which means that the overall skeletal muscle is going to get shorter. That pulls on the tendon, pulls on the bone, and that's how you move your body, okay? So that's the idea behind the T-tubules, and you know the deal. If that doesn't make sense, please leave questions in the comment section below, okay? All right, again, we'll come back to this idea of the terminal cisterns in a moment. Let's talk about a sarcoplasmic reticulum. Now that was on our muscle terminology slide. Remember, that's just an elaborate smooth endoplasmic reticulum. And in this little cartoon, the sarcoplasmic reticulums are these blue structures that are usually described as a loosely crocheted sweater, okay? All right, so if you look at the laser pointer, this is one sarcoplasmic reticulum, that's one. Here is part of a second sarcoplasmic reticulum. Here is part of a third sarcoplasmic reticulum. Okay, so along all of the myofibrils within a skeletal muscle fiber, you have many sarcoplasmic reticulums. Okay, the purpose of the sarcoplasmic reticulums is to store calcium. Calcium is going to be really important for myofibril shortening and contraction. And we'll talk about the role of calcium in future slides. But all of these sarcoplasmic reticulums are storing calcium, okay? And if we look at one sarcoplasmic reticulum, we'll take this one right here. If you look at the laser pointer is on the end of this sarcoplasmic reticulum. If I move the laser pointer over here, the laser pointer is now on the other end of the sarcoplasmic reticulum. So each sarcoplasmic reticulum has two ends, right? 1, 2. The end of a sarcoplasmic reticulum is a terminal cistern. If you leave off the S, that's the singular form of the word. So each sarcoplasmic reticulum has two terminal cisterns. We're going to put an S on it. So this is a terminal cistern, and that is also a terminal cistern. So there's two terminal cisterns. Now this is one of those terms that does have multiple spellings where they can stick an A on the end to make it singular, We're not going to worry about the different spellings of terminal cistern or cisterns. Let's just use this spelling, okay? So if you have to label this particular image on exam 3, you just label it terminal cistern. Let's just leave the A off, okay? But there are multiple ways to spell this. Okay, so then T-tubules are invaginations of the sarcolemma that run between each pair of terminal cisterns. Well, yeah, if you find, a pair of terminal cisterns that belong to two different sarcoplasmic reticulums, right? This is one sarcoplasmic reticulum's terminal cistern. Here's another sarcoplasmic reticulum's terminal cistern, and then the T-tubule is running in between them. The two sarcoplasmic reticulums are not touching each other. They are not touching each other, all right? There's a T-tubule that runs between them. All right, so then if we come down here to triad, tri is a prefix that means 3, and then you've got a bracket here. So the triad is a pair of terminal cisterns, one terminal cistern, here's the second terminal cistern, and then a T-tubule running up the center. That anatomical structure is the triad, two terminal cisterns with a T-tubule running in between. The triad is going to be important when we get to the physiology, okay? Here's another triad, here's a third triad, a fourth triad, 5, 6. Hopefully that makes sense. All right, so then when we get over here to the myofibril, you see these red and purple lines. These are going to be thick and thin filaments that are going to be present within the myofibril, and when we get to the next video, we will talk about those thick and thin filaments in more detail, okay? But for everything else, T-tubules, I think that's pretty obvious. We've covered everything else in this particular image. All right, thanks guys.